Okay. So very warm welcome and good afternoon to you from the Faculty of Pharmacy, Dr. Vishwanath Karad, MIT-WPU University, Pune. So on this 8th day of finishing school, I, Dr. Anil Pawar, Associate Professor of School of Pharmacy, take this opportunity to welcome the speaker of today's session, Dr. A. Van Dooren. I also welcome the Dean, Head, and faculty members of School of Pharmacy, coordinators of this finishing school program, and all participants for this session. Now we all know that no drug will get approval from regulatory authority without having sufficient evidence of its safety and efficacy profile. The potential drug candidate has to successfully clear all required preclinical and clinical investigation. So these clinical trials are usually conducted in phases, and each phase is designed to answer certain questions. Therefore, we need to have in-depth information about clinical trials. So with this background, today we are very fortunate to have today's speaker, Dr. A. Van Dooren, who will throw some light on this topic, that is why clinical trials. Through this topic, he will guide us on the importance of clinical trial in new drug development process. So Dr. A. Van Dooren is a PhD M.Ed by qualification. He completed his PhD in 1982 from Groningen State University. In 2002, Dr. A. Van Dooren completed a Master of Education from Tilburg University of Applied Sciences. He has extensive work experience in various fields such as pharmaceutical development, GMP, GLP, sales, etc. He also worked as a commercial director for medical disposable, cosmetics, food products, and pharmaceuticals in well-known organizations. From 2002 to 2011, he was professor of pharmaceutical business administration at Utrecht University of Applied Sciences. Dr. Van Dooren has experience as a research professor in pharmaceutical innovation and clinical research at Utrecht University of Applied Sciences. Currently, he is working as a trainer in clinical research and business administration at Pharmaceutical Solution, Rotterdam, Netherlands. He is also associated with Netherlands Pharmacovigilance Center. Dr. Van Dooren has over 60 papers to his credit. So I again welcome Dr. A. Van Dooren with the virtual bouquet as a small token of love from School of Pharmacy. I request Satish sir to present him virtual bouquet.
Okay.
Sir, we will welcome you, sir. We are welcoming you on behalf of School of Pharmacy, MIT-WPU University. Sir.
Thank you.
Okay. I now request our today's speaker, Dr. Van Dooren, to proceed ahead with his deliberation. But before that, I have a small announcement that all students are requested to switch off their mics, put the mobile phone on vibrator mode. In case you have any question, drop them in the chat box. We will have a question answer session at the end. You can also share your live feedback if you want to do so. Now I request Dr. Van Dooren to guide us. Over to you, sir.
Well, thank you very much for this kind introduction, and I'm very pleased to be with you today. Yeah, hopefully I can enlighten you a little bit on clinical trials in general, and specifically why we need clinical trials and what that would imply. For instance, if you think about it, more than a year ago, the world was got into the contact with a certain virus, and everyone at the time, we're talking about March 2020, was of course wondering why don't we have a vaccine? And since then, all sorts of clinical trials have been carried out in order to get a vaccine. Also, clinical trials have been carried out in order to check whether certain products would be effective against the disease. But it took some time, and as a matter of fact, it went very, very quickly, as we will see later. But clinical trials are needed in order to understand why a certain product, which at the origin may have a positive effect in terms of efficacy and in terms of safety on a certain disease, is indeed effective. And that's why we need clinical trials. So if I go to, let's see. Yeah, can you see this? Okay. Let's first go to standard care. This is a quote about, well, 5,000 years ago. And the quote states that superior doctors prevent the disease. A vaccine, therefore, is superior medicine, according to this quote. Mediocre doctors treat the disease before evidence of symptoms. And inferior doctors treat the full-blown disease. Most of doctors here in Western countries only come into action if a patient comes to them and tell them, well, I have this and this symptom, and what can you do about it? And they will prescribe a medicine or something like that. And this Chinese guy would say this is a type of inferior medical care. But what about doctors who just treat the symptoms? That's even worse, of course. This is about standard care. And now I'll go to the next one. Yeah. So are there any differences between clinical trials and standard care? Yes, there are. There are many differences. For instance, in terms of when we speak about standard care, we speak about patients. And in trials, we sometimes speak about patients, but not all the time. Usually we call them subjects or participants, but you can imagine that a trial on healthy volunteers, well, we can't talk about patients right then. So then in that case, we talk about subjects. Yeah. In trials, we focus on interventional effects, which are not known at the start. Yeah. We have extra tests during clinical trials. And we need to have methods to measure the intervention. And we have to have these measures right from the start, which means we need a protocol. So a protocol is an absolute requirement for a good clinical trial. The effects of the test will be measured over a period of time. So it is not a one spot at the moment. No, it is. Oh, sorry. Yeah, it is about a longer period of time in order to check whether the product is safe and effective. And we always will compare a group of subjects with another group. So there must be a comparison, a control group, and that control group can be on placebo, or they can be on the standard care. For instance, if you can imagine that the original tests on a COVID vaccine, when there was no vaccine available at the time, had to be done against placebo. But now the case is that there are certain vaccines available. So if at the moment a company would like to test a new vaccine, they can only do that against a standard vaccine that is already on the market. Because if you would do it on placebo, it would not be ethical. Standard care is about clinical judgment. That means it's flexible. If one prescribed product doesn't work, the doctor will try something else. In the case of clinical trials, that is not the case. There is a protocol, of course, within clinical judgment. If you see that a patient or participant is doing worse, then the doctor, it is the duty of the doctor to stop the trial and get the patient or the subject out of the trial. But for the rest, the protocol has to be followed. Now, before we go further, we first have to.
So, Dr. Dooren, sorry to interrupt you. Can you make your full screen? Can you make your PPT full screen?
Sorry, can I make?
Full screen, full screen.
Full screen. Oh, okay.
Yeah.
So press F5. Press F5.
Press F5.
Yeah, that is.
Yeah.
Yeah, and then it's.
Okay, thank you.
Yeah,
yes,
okay.
Thank you.
Yeah.
Thank you.
Okay. Oh. Yeah, so let's go to some definitions. I'm afraid we have to have these definitions first, because we need to know what we're talking about. Now, the shortest definition that we have on a clinical trial is the one mentioned here. A prospective study comparing the effect and the value of an intervention against a control in human beings. That's exactly what I already said. This is the shortest one. Prospective means that conclusions will be drawn that have a bearing for the future. So it is something you will check, and the conclusions will be used in the future. Now, there are also longer definitions, and here's one, which is done by ICH. ICH is the International Council of Harmonization, which is a collaboration between regulators on the one hand and pharmaceutical companies on the other hand, in order to reach quality improvement and to reduce duplication of testing. And E6 is about good clinical practice. So the ICH definition focuses on products, because this is about regulators and pharmaceutical companies, and they're more interested in products. So it's not only intervention, no, it is on products. And the definition is that any investigation in human subjects intended to discover or verify the clinical, pharmacological, and/or other pharmacodynamic effects of an investigational product, and/or to study what we call ADME. ADME is absorption, distribution, metabolism, and excretion. And the focuses in this definition are on efficacy and safety. Does the product work, and is it safe? They don't make a difference between clinical trial and clinical study. However, in the next page, I am giving you another definition, which is a definition by what we call the Ectr. Ectr is the European Clinical Trial Regulation. So that's a regulation in Europe, which is about what is needed on clinical trials. This regulation was issued in 2014, so 7 years ago. But it is still not officially effective. Now, hopefully it will be effective in the beginning of 2022. There are certain reasons for that, but most companies doing trials in Europe at the moment already rely on this Ectr, this regulation. And they make a difference between clinical study and clinical trials. A clinical study is an investigation in relation to humans. Clinical means humans, human beings, yeah, to discover the pharmacological, pharmacodynamic, or clinical effects of product. So it is, again, product-based. So that is about the effect. B, any adverse reactions. That's about safety. C is about the pharmacokinetics again, ADME, with the objective of ascertaining safety and or efficacy of medicinal products. So that's the definition of a clinical study. Now, a clinical trial is part of a clinical study. It means that participants need to be assigned to one group. And the decision about that is taken in advance. Yeah, so it is in advance in the protocol. It is already stated that there will be 2 groups, or even sometimes more groups, and that half of the subjects will be assigned to one group and the other to another group. The administration, the way the product will be administered, is also described in the protocol. Is it taken by capsule or by tablets, at what time, etc., etc. And there will be extra tests needed. So a clinical trial means that extra tests will be needed that are not needed in normal standard care. So this is the definition in the ECTR, and it's quite a complex definition. Like I said before, there are easier definitions, and here's another one, and this is the United States definition. It's quite recent, which states a research study in which one or more human subjects are prospectively assigned, prospective again, so it has a conclusion, the conclusions have a bearing on future care, prospectively assigned to one or more interventions, which may include placebo or other control, to evaluate the effects of those interventions on biomedical or behavioral outcomes. For instance, this is a definition which also would apply to the trial of a COVID vaccine. And here's another definition. Again, like I said before, we have to go through definitions before we can go on other things. And that is an important definition on what sort of product we are testing. Officially, a product that is still in clinical trials is not a drug product. The term drug is only used when the product is registered and on the market. So we call it an investigational medicinal product, or IMP. And an IMP is a product that is investigated or the reference. So a placebo is also an investigational medicinal product because it is part of the investigation, part of the trial. So the comparator may be a control already on the market, or it may be a placebo. And this also holds, for instance, for new indication or new types of patients. At the moment, there are trials on COVID vaccines for adolescents, children in the age of 12 to 18 years. And that is a new sort of population, which is also which also needs clinical trials, and which therefore the vaccine in that particular trial is called an IMP. Yeah. So this is the next one. It's also possible to have placebo. Placebo can be used as a reference. It's called an IMP, an investigational medicinal product. But of course, you can imagine that placebos may not always be ethically acceptable. Like I said before, if there are already vaccines on the market, then it is not acceptable to have a placebo when you are testing new types of vaccines. So why do we need clinical research, clinical trials? Well, simply to collect data on standard, usual, or also unusual events, and in different population groups, adolescents, for instance, in order to test a hypothesis. So, and the hypothesis usually is that our product is better than standard products, or is equally active and maybe safer. And that's the hypothesis that we're going to test in clinical trials. And the general goal is to better understand pharmacological processes, efficacy, and to improve treatment strategies in future. Oh, wait a minute, this one, yeah. So the objectives of clinical trials, the main objectives is to compare two or more groups of subjects using two or more treatment options. And a well-planned, well-executed, and properly reported and published clinical trial can be a very effective experimental approach towards evaluating the efficacy and safety of an intervention. So it's all about efficacy and safety. Does it work and is it safe? Yeah. And the intervention can be a certain product, an IMP, but it can also be a behavioral activity. If you want to study how well patients are taking their medication, how compliant they are with taking their medication, this is it's a behavioral study, and that's also a study that has to be done under clinical trial conditions and according to GCP, good clinical practices. There are many trials running at the moment. The 275,000 is a figure that I read about 2 years ago. I don't know what the figure is at the moment, but it's certainly not lower. And sponsors, so organizations that are starting such clinical trials, can be governments, can be universities, can be private institutes, health institutions, but also, of course, pharma companies that are interested in their product, but also device companies. So medical devices also need to be, many medical devices also need to undergo clinical trials, and that's another activity in which clinical trials are necessary. 15 to 20%, so 1 in 5 of all doctors in hospitals are involved in clinical trials. That's quite a lot, yeah. Which means that if you want to start a new clinical trial, yeah, it will be, yeah, it can be quite difficult to find a doctor who can carry out that study for you. So if you're a pharma company, you need doctors in hospitals to carry out the trials. But most doctors, and of course particularly the ones who have experience in good clinical practices, they are already involved in clinical trials. And yeah, the question is, can they have another trial, your particular trial, yes or no? So this is something that we have always have to take into account. But as you can see, 275,000 clinical studies, that's really a lot. So there are lots of studies carried out, not only on pharmaceutical products, but also on all sorts of behavioral health effects. Here are some current developments on clinical trial. First one is adaptive licensing. This means that the marketing authorization is given for a new product, is given provisionally. So, and during normal use on the market, further data will be collected and will be submitted to the regulators on a regular basis, which could subsequently lead to definite approval. Or you could start with a smaller number of patients for the product on the market, then accumulate the data and increase the target groups. That is what, for instance, has happened with COVID vaccines in Europe. They got a provisional license, and under the condition that data on the safety and the efficacy had to be collected. And based on these accumulated data, later on a full swing license or authorization can be given. Genomics and personal medicine. Genomics is a field of biology that focuses on the structure, function, evolution, mapping, and editing of genomes. One of the questions that is still, again, I know that you in India are also very much involved with all the problems of COVID. So I'm using COVID as an example, but of course you can use another example too. But for instance, one of the important questions is, why do some people need oxygen? Why do some people need to go to the intensive care? Why do some people die on COVID and others just feel it like a common cold? Yeah, that probably has to do with genomics, and we still don't know the answer. And of course, as soon as we would have an answer to that, we can, of course, be more specific on how to prevent the serious consequences of COVID to happen in the first place. Fail fast, fail cheaply actually means for pharmaceutical companies don't spend money on products of which you already know that they are inferior to what is on the market, or they have a weak spot. So as soon as something like a safety side effect, an adverse event would come up during clinical trials, that usually for companies is the reason to stop the development altogether, because it will never get better than what is on the market already. Focuses on processes and training, I think that is self-explanatory. More stringent post-marketing requirements. The term post-marketing actually is a bit strange to me, because post means after, and after marketing would mean that the product is taken from the market. So it's at the end of its lifetime. Post-marketing usually means post-authorization. So the product is authorized, it's licensed to have it on the market, and there are certain requirements with respect to what we call pharmacovigilance. So that is the collection of data that is needed to get more information on the safety of the product. Yeah, and these data have to be added to the regulators in order to get a better overview. So side effects of COVID vaccines have to be accumulated during the phase of marketing. And of course, more frequent and intensive inspections, FDA, EMA. FDA is the Food and Drug Administration in the United States. EMA is the corresponding authority in Europe, which is called the European Medicines Agency. They work together, and that is also part of globalization. Globalization means access to more investigators and subjects, access to new markets. Yeah, studies being done in Japan or in Vietnam or in India, which have already been done in Europe, gives information on whether populations, different populations, also respond in the same way as we would have expected from earlier studies. Larger population, especially for pragmatic trials. Spread of ethnic groups and a broad spectrum of diseases. That is something that is also something that is the result of globalization. So clinical trials are done all over the world. Here you can see this is a picture taken from an FDA site on globalization on clinical investigators or clinical trials. And what you can see here, red is trials in the United States, green is in Western Europe, and black is in the rest of the world. And over the years, you can see that the number of trials in the rest, what we would call the rest of the world, those parts of the world which are not United States or Europe, is increasing heavily. Does this mean that, for instance, in the United States, the red one, the number of trials is decreasing? Well, not exactly, because what you can see here is that the number of trials explodes. So also in the United States, although relatively the number is decreasing there, in actual size it is still growing also over there. So the number of trials is really exploding. And that also holds for the pharmaceutical market. So the market of pharmaceutical products is also exploding. This is a picture before COVID, and I think that also when you would include the COVID data in it, you will see the same line of the error, that the pharmaceutical market is increasing. Yeah. So pharma has always been a growing market. I have to add that this data are in money, and of course the pharmaceutical market also has to do with price issues. medication is expensive, and with patent issues. I'm not going into details with that, but that would be something if we would discuss the ins and outs of the pharmaceutical industry and big pharma. But of course, price and patents are important issues when you look at the pharmaceutical markets in total. Drug development costs are increasing as well, and that's something you can see here. And these are data which show that the total amount of money you need for developing a new pharmaceutical product amounts to $2.5 billion US dollars, of which about 40% is non-clinical and 60% is clinical. Now, there are some persons who think that 2.5 billion is really exaggerated. It is too high. The lowest figure I have seen was in the region of 1 billion. Yeah, still 1 billion for a new pharmaceutical product is a lot of money. This does include the amount of money that you will be spending on products that will never make it. So products that during the clinical trial phase are shown to be inferior because they don't work or because they have safety issues. Like I said, fail fast, fail cheaply, will be taken out of development, but the cost will have to be borne by the products that are on the market. Yeah, because somehow it has to be paid. And this also is included in the 2.5 billion, which is quite an amount of money. Some people, for instance, here in Europe, they say, okay, well, why not, because of the high pricing of pharmaceutical products, why not assign the task of drug development and the development of new drug products to national the European committee, for instance. Yeah. Well, the problem then is, well, is it ethical to spend 2.5 billion US dollars on products that may not come to the market? So this is something that has to do with the development and the use of products and development cost, which at the moment is still based on pharmaceutical companies. So here we see something about the phases of clinical development of investigational medicinal products, IMPs. And what you see here is that what we call a phase 0, that's effect on the body, and phase 1, phase 2, phase 3, phase 4. And what you see that during these phases, the number of subjects, the number of participants is increasing. Yeah. Also, the duration of the trial is also increasing. Safety in humans, which is done on volunteers, healthy volunteers, is something that takes about usually a couple of weeks. Yeah, but the effectiveness of the treating diseases, so in patients, is a matter of months. Phase 3 is the large-scale safety and effectiveness, and that is on thousands of patients, and it can take years. So what we have seen in COVID vaccines, that this has been that this was done within, well, something like 8 months, is extremely fast. This is really something that gives an insight in how important these trials were and how quickly these trials were done. Long-term safety is something that is done when the product is on the market, and that's what we call phase 4. So these are the various phases, all having their own conditions and requirements. So now let's go to a little bit of history. The first controlled clinical trial that has been described, I must say, was a trial by the Holy Roman Emperor Frederick II of Hohenstaufen. And you see a picture of him here. He lived from 1194 until 1250, so some time ago. And he was really, science-driven, you could say. And one of the things he wanted to know is digestion of food faster when you do nothing, when you sleep, for instance, or when you are very active? And what did he do? He took 2 knights out of his courtroom and he fed them with lots of food. They could eat as much as possible, and they had to eat as much as possible. Then he sent one of them to his sleeping room, and the other one he sent on hunting. And after a couple of hours, he got them back in his courtroom. So the one who had his food, he was that was group 1, and he slept. And the other one who went into activity and went hunting. The next thing what he did was he cut their throats. And then he opened their bellies and he looked into their guts and checked in order to check whether the digestion was better in one of the groups. And I can tell you that what he found was that the digestion was faster in the sleeping night. So that was his conclusion. Now, this was a certain study. My question to you is this controlled clinical trial, was it ethical or not? Well, I can tell you it was unethical, but here are some of the reasons that you could state about why it is unethical. It did not include an IC process. IC means informed consent. So you ask the participants, are you willing to participate in this particular study? And you can only do that if you tell them what it means. I think that if I would have been a knight and I would have given the opportunity to say yes or no to the trial, knowing what the results would be to my throat, I would probably have said, no, I would rather not. Yeah, but of course they had no choice. Participation was no voluntary. Yeah, it was not randomized. Yeah, the statistical power was insufficient. Yeah, the intervention was improperly designed. There was no protocol or something like that. This was what he did. The outcome measurement was not defined properly. Yeah, and the resulting event was the subject's death. So my question to you is, which of these reasons do make this trial unethical? And here's the answer. All of them. There was no IC process. It was not voluntary. It was not randomized, which means that he said to the 2 knights, you are going to hunt, and to the other one, you're going to sleep, rather than do this blindly. Yeah, the statistical power was insufficient because he only had 1 participant per group, which does not give a very strong statistical power, of course. It was improperly designed because normally you would say, okay, we need to know how much they will eat. We need to know, are they going to sleep indeed, and how active will they be during hunting? Yeah. So it was not standardized in that sense. The outcome measurement was not defined properly. It just got their bellies open and then said, well, in this particular case, I think that digestion is better. But how to describe that is something that he should have done beforehand in a protocol. And of course, the subject death is something that is unethical as well. So this is one of the first trials that has been described. But let's go to another one, which may be a bit better also in terms of its ethical nature. This was a randomized trial, and James Lind, the person you can see here on the left, was a doctor in the British Navy. And one of the things that he had to deal with was scurvy. On long boat trips, the people on board sometimes got agonizing pain, which could even lead to death. Their teeth fell out, and it was an incredible disease, and they didn't know what the cure was. Nowadays, we know that this has to do with a lack of vitamin C. That's why it's called scurvy. It's a lack of ascorbic acid. Yeah, but at that time they didn't know. So what he did was he carried out a trial in which he used 12 sailors, and he randomized them in groups of 2. Group 1 got 1 quart of cider. Group 2 got 25 drops of sulfuric acid solution. Group 3, 6 spoons of vinegar. Group 4, half a pint of seawater. Group 5, 2 oranges and 1 lemon. And group 6 got a spicy dish plus a glass of barley water. And what were his results? Let me first say this was not blinded. So he just picked 2 sailors for group 1 and 2 sailors for group 2. It was not blinded. But he got an astonishing result. And that result was that group 5, the group with the lemons, got a huge improvement. They recovered, actually. Unfortunately, he had to stop after about a week because he had no fruit left. And after that, the sailors, they didn't get any ascorbic acid, as we know now, and they died anyway. Yeah, which at the moment we would say is unethical. If you start a study, then you should ensure that there will be enough IMP available for the total duration of the study. Also, the group that got the cider found a small improvement. He did not, unfortunately, realize what the consequences of his trial were. It was only a footnote in a book that he wrote 6 or 7 years later, and he did not implement it. Only in about 50 years later, the routine use of lemon in the British Navy was started by Admiral Cook. And since then, sailors in the British Navy are called limeys because they needed to take to have lime or oranges. As a matter of fact, it is strange enough that the Dutch that were fighting the English in the 17th century and also had a big fleet, they did much better because they grew scurvy grass, Cochlearia, on their ships. And sailors had to eat the leaves of scurvy grass. They didn't know that these leaves contain a lot of ascorbic acid. That was something that was only later on found out. But it did help to prevent scurvy. Here's another one. It's Ignas Semmelweis. He was a Hungarian, and he worked. He was a gynecologist, and he worked in the famous university clinic in Vienna around 1850. That was a clinic of which Professor Klein, he was one of the main major gynecologists in the world at that time. But Semmelweis noticed that very often after giving birth in that university clinic, women after about 24 hours developed a pain, excruciating pain. It is called puerperal fever. And after a week, they died in really great pains. And what he considered was that these fever incidences occurred much more in the university clinic than in a neighboring clinic of nuns in a convent. So those women who went to that convent had a much better chance not to develop puerperal fever. And he came to the conclusion that it had to do with the cleanliness, because in those days, a good doctor could be shown on the amount of blood on his white coat. So the redder his coat, the better it was. Yeah. So they came, for instance, after doing trials on animals, they in their same lab coats, they went to the woman they needed to help giving birth. And he said, well, maybe if people would wash their hands, that could be helpful. So the doctors, he ordered doctors in the university to wash their hands, which was quite unpleasant because that washing had to be done in it was very, very unpleasant because it had to be done in glory. Yeah, and that is rather unpleasant. But he found that by hand washing, he could decrease the mortality to 3% of the original figure. Yeah, there was no proof, and he had no idea that this had to do with bacteria. Remember, we're talking about 30 years before Pasteur. And the mistake he made was that he tried to implement it, but he did it on a very, not a very good way. He accused his Professor Klein, the world famous gynecologist, of being a murderer. Well, that is not something you should do to your boss. One of the other things was it was no proof, because the question was, if you decrease it to 3%, why not 0%? Yeah, well, that might have to do with the way of washing, etc., etc. So he was sacked. As a matter of fact, he went back to Hungary, and he died with a couple of years later with symptoms similar to puerperal fever. So he might have contracted it himself. It did not occur to him that the practice of surgical profession needed to be changed, and he did not recognize the role of germs. Okay, now let's go about a century further on, around 1950, and there we have Austin Bradford Hill. Austin Bradford Hill was an English guy who wanted to become a doctor, but he got TB, tuberculosis, and that was the end of his career as a doctor. Instead, he concentrated on what we now call medical statistics. He carried out a study on streptomycin, that was one of the, at the time, newer antibiotics, and he tested these in TB patients. And he did this in a randomized blind way, and he introduced statistics. So he checked statistically whether the product worked or not. He also did a certain study on smoking and lung cancer in the 1950s. And he found out that there is a direct link between smoking and lung cancer. He presented these data to the English government, but the government at the time said, well, we don't want this information to be disseminated, to be made public, because we need the taxes from the cigarettes. So that was at the time considered ethical. He found out that, can I get, oh no, I have to do that myself. Let me see. Yeah, he found out that you can level scientific evidence. That if someone says that something is true, we call that a Delphi, an expert opinion, or if one person is recovered from a certain disease by means of an intervention, case report, that is of course some form of scientific evidence, but it is not a very hard form. the best form of getting scientific evidence is a systematic literature review, particularly those in which all studies on a certain interventions are combined together, what we call meta-analysis. We can also have randomized controlled trials with hard results, or randomized controlled trials with non-descriptive or non-decisive results. For instance, if you want to check whether an antihypertensive is effective, actually you should wait until people will develop a brain hemorrhage, which of course is not ethical. If you go, if you continue the study that long, that some of the people will develop brain hemorrhage, that would be not ethical. But that would be the hard result. A non-decisive result would be a blood pressure measurement, yeah, which therefore is a little, yeah, it's less hard, but still okay. Cohort studies are studies in which a population at risk of getting a certain disease or symptom is followed over time. So at the study start, no one is ill yet, but you wait, it can take years and years and years until certain proportions of the population have developed that particular disease. It's quite expensive and it's time consuming. Observational studies means that you draw inferences from a sample to a population, where the independent variable is not under your control because of ethical concerns, for instance. For instance, the possible effect of a treatment of subject when you are not able to assign the subjects to group. And cross-sectional studies are studies in which the data are collected from a population at a single point of time. So the Holy Grail at the moment, and that is really based on the information and the studies that Bradford Hill carried out, is what we call the randomized controlled trial. And that's what we call evidence-based medicine. So the last picture I gave to you is what we call evidence-based medicine, which means the conscientious, explicit and judicious use of current best evidence in making decisions about the care of individual patients. So you collect all data, also from medical publications, etc. And these data then are being weighed together with your expert opinion as a doctor in finding out what the best care for your particular patient should be. And this is what we call the Holy Grail. So randomized controlled trials are still the Holy Grail, but what you will see that in future is that other types of trials will also get their place in clinical research. So it was Archibald Cochrane who was the person to develop evidence-based medicine, and you see the same picture here. Meta-analysis is the top, that's the best clinical evidence you can get. Then you get randomized controlled trials, cohort studies, case control study, case reports, and then IDs. Yeah, and animal research is still below that. So this is what you would call evidence-based medicine, a hierarchy of evidence from trials. Now, what is important to realize is that statistical significance is not the same as clinical utility. And sometimes the actual real world can be different. And this is, this is Snow White, a beautiful picture. Everything is okay, but the actual world may be different. And this is what could be the case later on, of course. And the prince who saved her, appears to be someone who was sitting, not doing anything on the education of the children, etc., etc. And this actually is something that we have to take into account in all cases of randomized control trials, because in randomized control trials, what you would like to see is that the effect that you find is only due to the intervention. which means that you need to have patients who are not smoking, because otherwise smoking can get an effect on the results. They're not have another disease. They're not diabetic or something like that. They're not too old. They're not too young, etc., etc. So what you're actually looking is, you're looking for the perfect patient, and then you hope that the results will be such that they will also be valid in normal clinical practice. So there are some criteria for statistical relevance. If you find that a result is statistically significant, and you want to give that information to the real clinical world, it would be nice to see that the correlation is biologically plausible, that the correlation is strong. That the correlation must reflect a biological ingredient or biological gradient, so that if you see that the dose is doubled, for instance, the effect is doubled too. Yeah, that the correlation must be found consistently, so not in only one patient but in all patients. That it holds over time, and that the correlation is preferably confirmed by experiment. That is what Austin Bradford Hill gave us the medical world. Now, I have said lots of words already, and now it's time for you to have some questions and try to answer them. I have some questions for you, multiple choice questions, and I'd like you to find out what the best possible answer is. Okay, so I'm not asking for the only answer, because in this world, usually the more answers are okay, but I'd like to hear from you what the best possible answer is. So here's my first question. Randomization refers to, and there are 4 possibilities: ranking the level of scientific evidence, taking into account an ethical code of conduct, an unbiased allocation of subjects to experimental and control control groups, of ensuring statistical significance. I'll give you a couple of seconds to think what the best answer would be.
So participants, you can write your responses in chat box.
Excuse me?
Yeah. So student can write their answer in chat box.
Okay. Yeah. Yes. Okay.
Okay.
Okay. So shall I go to the answer then?
Yeah.
Everyone,
one student is saying option 3. Option 3 is correct.
Okay. I'll wait. Yeah. So 1, 2, 3, or 4. Okay. Yeah. And here's the answer.
Answer, students saying answer number 3 is correct. Sir, you please unmute yourself. You are not audible, sir.
Sorry.
Yeah. Now it is audible.
Yeah, I'm sorry. Yeah. Number 3 is the correct answer. It has nothing to do with evidence-based medicine. I explained that to you. Of course, an ethical code of conduct is very important in clinical trials, but that is not what randomization refers to. Randomization refers to the allocation of subjects, unbiased, and the subjects to the different groups. And statistical significance may be the result of a clinical trial. So that is not something that is the definition of randomization. Okay, shall we go to the next question then? So what seems to be the best randomized allocation? Is it number 1? Is it number 2? Is it number 3? Or is it number 4? A, B, A, B, A, B, A, B, A, B, 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 A, B, A, B, 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 A, or B, A, B, B, A, B. Yeah. What is the best randomized allocation? Remember, the best possible answer. Okay. So here's the answer. AB, 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 AB suggests that it is not randomized. Yeah, it is possible, however, but it is not. If you look at these possibilities, then number 3, where A and B are randomly divided, seems to be the best answer. So A, B, 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 isn't it? And B, A, B, B, A, B probably is not either. So look at the picture. There you see that randomization is all, it depends. You do not know beforehand in which group you will become as a participant, but also not as a doctor. Yeah? Okay. Next one. One of the main safeguards for doing clinical research is 1. Asking for permission from an IEC, IRB. An IEC is an ethics committee, and that is a committee that beforehand, so before you are allowed to start, must give approval of the study in the particular hospital. So it is mainly hospital-based, and they check whether the product is ethically sound in terms of what you are going to do to the participants. EC is the European term. IRB, Investigational Review Board, is the American term. They're practically the same. There are very small differences between the two, but we don't have to bother about that. So the asking for permission is something that you need to do. So that's a safeguard. Informed consent is also a safeguard. I explained to you in the case of the second of Hohenstaufen that informed consent is extremely important. James Lind didn't ask for consent either, but at the moment we know that if you want to have someone included in your trial, that particular person must consent to it in a written form even. Yeah, that's what we call informed consent. An investigator with a CV showing extensive experience in GCP and study conduct. That's also something that is very important when you do the study. An investigator that has no experience at all in GCP, well, he still has a learning curve to go. Yeah, and solidarity with patients is also something that has to do with safeguards. So, what's your answer?
Many students are saying it's answer number 2, informed consent.
And that's very correct. Excellent. I'll give you an applause. That is the best, again, the best possible answer. Also, the others are important, yeah, but the best possible answer here is informed consent. And like I said, at the moment, we ask for informed consent in a written form, yeah. There must be written information on what the possibilities for the trial are in terms of how dangerous it is, what the likely outcome could be. But in the end, the patient has to sign a form in which he states that that he understands what it is about, and that out of his free will, he is going to participate in the trial. And that sometimes is quite difficult, because you can imagine that if you have a certain disease and the doctor tells you, well, I don't have anything, there are not treatments that I can offer to you except for a clinical trial. So would you be interested in trying that? Well, yeah, you don't have a lot of possibilities anymore. Yeah, but so the question is that free or not? And well, that's the very important ethical consideration. Okay, you have worked hard, so I suggest that we have a 5 minutes a break before I will go on with the second part of my talk.
Yes.
So 5 minutes. Okay.
Yes. Thank you, sir.
Okay.
So, student, you can take 5 minutes break. We'll meet again after 5 minutes.
Pawar
Satish
sir,
sir?
Pawar
Yes.
sir, Rutuja will take the questions at the end. Rutuja Vinsurkar. She will,
Okay.
yeah.
Okay, sir.
Yeah, Rutuja, please.
Perhaps you could give me a sign if we could start again.
Yes, we can start now.
Okay. Okay. Then we'll go to the next slide, which gives you some information on new developments. I already said that Of course, like we said before, the randomized controlled trial is still the norm, but if that would always be the case, we would have never set foot on the moon, because you cannot do a study beforehand in order to know whether it's possible, yes or no. So for instance, we have cluster RCTs, in which particular case you're not going to check individual persons, what is the standard of an RCT, but groups of persons, for instance, families or school classes or villages, etc. In adaptive design, you adapt, for instance, the sample size or the dosages of patient characteristics, or you stop the study earlier because of the data collected during the study. So in a standard design, you have the design, the protocol, then you carry it out, and then you analyze. In an adaptive design, you also have a protocol, but during conduct, you already review the data, and that could lead to a change in the characteristics. Yeah? Basket or umbrella trials, in that particular case, well, an example would be a treatment of certain forms of cancer, which have the same genetic basis. And this way we might have faster results. So it's not a type of cancer, but it's a genetic basis that you're checking. Platform trials, that means different interventions at the same time. We call it also MAMS, M-A-M-S, multi-arm, multi-stage. Then we have pragmatic trials, which means in real-life practice under normal clinical circumstances. So it is in contrast to the explanatory trials, in which, like I said before, you are looking for the perfect patient, the patient who has no other disease. Yeah. In pragmatic trials, you know that the persons in your study may have all sorts of other diseases too. In the new revision of the GCP ICH guideline, E6, pragmatic trials will be included, like also real-world data trials, in which you use cases, for instance, the collection of vaccine side effects. Decentralized clinical trials are defined as studies executed through telemedicine or mobile local healthcare providers. So that is processes differing from the traditional clinical trial model. And there also is a possibility of what we call NS1 trial. That's a single case study. If one patient responds well to a certain intervention, then patient number 2 is randomly and blindly assigned to either a reference or the same intervention. And that, and if you manage the outcome systematically, that can also be a possibility for new designs. Now here's a good pragmatic trial, and we're talking about 1954. It was the largest clinical trial that was ever done. Over 600,000 school children were injected with a new vaccines against polio. And there were more than 1 million children as controls. Yeah, so they didn't get anything. So the 600,000 were divided into half, approximately half for the vaccine and another half for the placebo. And then lots of children were used as controls. And what it was found that this polio vaccine was both active and safe. And in 1994, in the United States, polio became even extinct. So before that time, polio was it's a disease which could heavily affect the life of patients, children who got it. But now, fortunately, it is quite extinct. Although I have to add that the numbers, the incidences of polio are increasing again because there are many people who don't want to vaccinate. But fortunately, hopefully, this is something that is also going to happen with COVID. That in the end, people are becoming immune to it, and that's a sort of herd immunity that helps to stop the spreading of this event. this product was never patented. And when Salk was asked, why didn't you patent this polio vaccine? He said, can you patent the sun? Can you patent sunlight? And that's not possible either. So that's how we compared it. Okay. Now let's go a little bit to the rules and the regulations. We already talked about ICH. EMA is the European Medicines Agency, and FDA is the Food and Drug Administration in the United States. I also included the Japanese PMDA, the Pharmaceutical and Medical Devices Agency, and of course there are many more. Every country has their own agency for registration of drug products. So which rules should you be following? Well, fortunately, this is not the case. It's not a matter of heads or tails, totally not. But it certainly depends on where you are going to register your product, or in which country are you carrying out this particular trial. Yeah. Usually, pharmaceutical companies will look for a global registration. So not only in Europe, not only in India, but also in the United States, in Canada, in Australia, in Japan, etc., etc. And they have to comply, therefore, with all the registration procedures and conditions and requirements in those particular countries. So you can imagine how important it is that registration authorities look to each other and try to combine their registration requirements, so that if you do a test for a European for the European registration, that a test will also be valid in United States or India or wherever. A trial, a clinical trial, also has a life, and it starts at the conception. Then you have the pregnancy, you have the birth, you have the toddler phase, etc., etc. And in the end, a trial also dies. So here are a few of the different phases in a clinical trial. Yeah, the conception phase is the ID, the description of the research question. I want to know whether my product works, whether my product is safe as compared to standard treatment options. Yeah? Then we have an internal preparation phase, the budgets, the protocol, the project organizations, etc. And we have an external preparation phase. We look for investigators. We look for contracts with the investigators. We look for a CRO, a clinical research organization that will do the work for us in a certain country. Yeah, we arrange for submission to the ethics committees, to the IRBs, and we aim for submission to the registration authorities. Then we get the green light. That's the study start. Yeah. Then enrollment starts, and the adolescence. Then we get the adulthood, the study conduct, safety reporting, quality control, and oversight by the sponsor and the investigator. Then there is a maturity and retirement phase, the analysis of the data, reporting of the data, things like that. And then we have the study closure, the death. Yeah, that can be positive. The product works, is statistically safe, or, and that is mostly the case, unfortunately, it's negative. It doesn't work, or the investigator is losing interest. There's not enough enrollment. There's insufficient power, etc., etc. After the trial ends, we need publication or registration, and sometimes when the product is on the market, also pharmacovigilance, which means that the study of safety for products that have already been marketed. So clinical trials also have a life cycle. Some issues in clinical research, internal versus external validity. I gave you the example of Snow White. A lack of oversight. Oversight means that you are in control. That is needed in order to have sufficient patient protection, and to have better data quality. Difficult in record keeping is sometimes something that is that jeopardize can jeopardize the outcome of the clinical research. Costly reiterations. An example, for instance, of a reiteration is the AstraZeneca COVID vaccine. In the beginning, they found that they knew that they needed 2 shots, about 6 weeks, one after the other. But they found originally that after the first shot, the protection was better than after the second shot. That is not very logical. Remember Austin Bradford Hill. So they had to do another study in order to check what was wrong, and it came out that they misinterpreted the data, and that even some data were not recorded properly. Yeah, and that is of course quite costly. Reduced trust. Well, you can imagine that. Low patient engagement. Patients, you need patients to include them in the trials, to enroll them, and you are very much dependent on these patients. And also difficulty in finding the right partners. Like I said before, many investigators are already involved in the trials, and you want to have them also being involved in your trial. So that can be quite a challenge. Yeah. The ethics central maxim is that the interest of subjects should always take priority over all other interests. So priority, for instance, over interest in data integrity, of priority in interest of the safety of the people working within the trial. This is very important. It is mentioned in GCP, ICH E6, and also in what we call DOH, which is the Declaration of Helsinki, and that is also a sort of central ethical guideline on clinical trials, mainly used for doctors. Here are some of the key persons involved in clinical trials. The EC and IRB, we already discussed it. The DMC is the Data Monitoring Committee, and that's a sort of a committee that collects the data during the conduct, is even able to unblind them without the investigator or the sponsor knowing it, and come to conclusions on whether or not it is safe to continue the trial. Sometimes we also have a CTSSC, a Clinical Trial Steering Committee, particularly in cases of difficult, complex trials. We have a sponsor, that's the pharmaceutical company, for instance, who owns the product, who owns the patent, and who owns the money and pays for everything. Project managers, a CRO, a clinical research organization. If an Indian company would want to carry out a study in the Netherlands, for instance, they will probably not do that from India, but they will look for a local company in the Netherlands, which is focused on doing trials in our country. Yeah. Investigators, PI means principal investigator, but there are also other types of investigators, of course. A CRC is a clinical research coordinator, sometimes also called the study nurse. She is doing the actual work in terms of documentation and simple testing under the oversight of the investigator. We have the pharmacy. We have the pharmaceutical challenges are, of course, quite interesting here, because what you need to do is you need to have a product and a reference, both called IMPs, which are exactly the same. Yeah, they must have the same size, they must have the same color, they must have the same taste, things like that. It is not allowed that you can see a difference between the reference and the thing that you are testing, and that's quite a challenge for the pharmacy. So it is an interesting activity for pharmacists to do such a thing, and to prepare products for clinical trials. Laboratory, all the laboratory testing, etc. Data management. A CRA is a clinical research associate. That's the person who does the monitoring of the testing, checking whether the trial is being carried out according to the protocol, according to GCP, and according to the sufficient safety of the patient. And of course, you have the patients, or better called subjects. There's a lot of documentation in clinical trials, and many of these are called essential documents. And the definition of essential documents is those documents which individually and collectively together permit evaluation of the conduct of a trial and the quality of the data produced. These documents serve to demonstrate the compliance of the investigator, of the sponsor, and the monitor, the CRA, with standards of GCP, good clinical practice, and with all applicable regulatory requirements. So these essential documents assist in giving you the certainty that the product is done according to the protocol, that the study is done safely, and that the results will be credible. Yeah. So it will they will also assist in successful trial management, and they will be available for audits and inspection even afterwards. Yeah. They are regulatory documents. So if you want to register your product in a certain country, you have to provide the regulatory authorities with these documents. So quality assurance is very important, and this is the definition of quality assurance. All those planned and systematic actions that are established to ensure that the trial is performed and the data are generated, documented, that is recorded, and reported in compliance with GCP and the applicable regulatory requirements. So it is systematic action, and that's what we call quality assurance. Apart from that, we also have quality control, and quality control is the operational technique undertaken to verify that the requirements for quality of the trial-related activities have been fulfilled. So every company, every organization has a quality management system, of which quality assurance is a part, and quality control is part of quality assurance. That's how you should divide these different quality words, actually. Okay, now here's some annex, and that has to do with career options in clinical research. A CRA is a clinical research associate, and that is someone who is doing the quality control, so the monitoring of the testing during conduct. And they check whether everything is done according to protocol, whether the data are credible, and whether the patients are not in jeopardy. Most of the time, people start as a CTA, a clinical trial assistant. And then we have an in-house CRA. And only later on, people then go to the hospitals themselves in order to do the testing and the verification, the monitoring. That's what we call a normal CRA. A senior CRA is someone with lots of experience, and a senior CRA can grow into managing a number of CRAs for a certain test or for a certain trial. But they can also go into project management, they can also go into QA management, and they can go into pharmacovigilance or safety. And it is also possible for a CRA to start for themselves, to start on their own. That's what we call a freelance CRA. And sometimes freelance CRAs are asked for to do testing or monitoring for certain studies, and they are assigned by pharmaceutical companies. The outlook for careers in clinical trials are extremely well. And the 10%, which you can see here, is when you talk about increases in salary, then 10% is sometimes even on the low side. You can become a registration officer. You can become a registration director. You can work in data management, in medical writing, in market access, or HTA, health technology analysis. And that is about the efficiency of a new clinical treatment option. That has to do with the pricing. Yeah? Project management, auditing or quality assurance, contract management, the contracts between sponsor companies and investigators, new business development. So that's more the sales side of it. Clinical management, you can work in pharmacy, laboratory, or you can even become a medical director. Yeah. And a medical director, by the way, is someone that is, the medical director of a company like Pfizer in the United States is earning something like $400,000 a year, excluding bonuses, etc., etc. So, yeah, not too bad. Okay, one last question then. Again, I'd like you to to spend a few moments to try to answer this particular question. What is the prevailing consideration when mitigating clinical trial management to cope with COVID constraints? Imagine the situation last year when there were lots of clinical trials being conducted in hospitals, and then suddenly these hospitals said, well, we can't do it anymore because all our investigators now are need to work in the IC to help COVID patients. The ICs and hospitals were flooded with patients, a situation that you are aware of. And they said, well, at the moment we cannot continue this trial. So you have to do something about it, sponsor. Now, what is the most important consideration here? Is it the safety of the current participants? Participants who were already in this particular study. Was it the validity of the data? Was it studies on potential COVID remedies that will have always priority over standard trials? So a trial on a COVID vaccine or a possible COVID medication will start earlier and the standard trials will be stopped? Or is it the protection of the healthcare workers involved in trials against COVID contagion? So healthcare workers that should be safe from COVID contagion. Now, which is the main, again, best possible answer, which is the main consideration when you are in such a unfortunate situation? So take a few moments again to try to answer this question.
Yes, participants, you can put your responses in chat box.
Ready?
Option 4. One response is all of them are equally important.
Option 4 is what most people say.
Yeah,
well,
option 4.
Yeah. Okay, well I don't agree. Because I think it's a trial participant safety. So the safety of the participants, remember what I said about DOH, that is the most important thing. And the rest is also important, certainly, but the safety of the patients, the safety of the persons that are included in the trial, that have already been enrolled, that's the number one consideration which we take into account. Yeah. So even if the validity of the trial data would be jeopardized, trial participant safety is the main thing. And that can mean that the trial has to be continued. But for instance, you don't ask people anymore to come to the hospital because safety in the hospitals was not 100% guaranteed. So sometimes participants in a trial were asked to do all sorts of testing at home, and the study nurses went to the homes of the patients in order to do these testing, rather than asking the patients, what was the normal situation, to come to the hospitals. And that is something that has to do with trial participant safety. So yeah, that is still the most important thing, and it will always be. That's the central maxim. And as a matter of fact, this will conclude my my talk. So my question then is: are there any questions from your end that I might be able to answer?
Yes, thank you, sir. So now it is a time of question answer session. So I request participant to drop the question in chat box.
You can unmute and you can ask, no problem.
Yeah. So any question, any query? Satish sir, can I proceed further?
Yes, sir. You can. Yeah. Continue.
Okay. So now it's time for live feedback. So I request some of participants to share their live feedback.
Aditi Kole is there. Aditi Kole, please can you give your feedback?
Hello.
Yes, Aditi, you're audible. Please go ahead.
Yes. Yes, I want to say thank you because we got many more insights in the clinical trial session, in the clinical research part, and it was a great learning for today. And thank you so much. And also, I want to thank MIT World Peace University for giving us opportunity to join the session. Thank you.
Thank you. It was my pleasure to be with you today.
Yeah, sir, one more Nitin Naik is there. Can you, Nitin, can you give your comments, live feedback? Yes, Nitin, please.
Hello, sir. Myself Nitin Naik from Shivaji University, Kolhapur. I'm a research student in Shivaji University. I'm also working in a clinical field. So it's a great pleasure for me to be a part of this workshop organized by MIT World University. And thank you especially, sir, for you because you have mentioned very simple examples in the history as well as in current scenario. And you have touched all the points in your lecture. So it's a great pleasure to listen to you, sir. Thank you from Shivaji University to MIT and also to you, sir. Thank you.
Thank you, Nitin. Thank you for sharing your views.
Yes, sir. Pawar, sir, you can proceed.
Yeah.
Pawar, sir, there is one question in the chat box. Can I take it?
Yes, please.
So the question is, can this COVID have impact on clinical trial as everything is remotely going on? Adoption of remote monitoring technologies can decentralize approaches.
Wait a minute, I don't think I understand the question completely. Could you repeat it, please?
Can this COVID have impact on clinical trial as everything is remotely going on? Adoption of remote monitoring technologies and decentralized approaches.
Yeah, remote monitoring is one of the newer approaches, actually. Well, certainly due to the COVID experience, remote monitoring was the only possibility for CRAs to check the conduct and to verify the data and whether or not everything went okay. In my opinion, remote monitoring is certainly helpful, but it can never be a complete substitute for personal observance. I am sure that when this pandemic will be over, remote monitoring is there to stay, but going to the hospital and visiting the hospitals and the places where the trial is being carried out is still something that is going to be done again. I think that what people then will find is that remote monitoring again, will be helpful, but it may not be 100%, and therefore, the monitoring on site is still something that will continue to be done. Is that an answer to your question?
Yes. Yes. So is there any other question? Okay. So thank you, Dr. Van Dooren, for such an informative session. In your session, you clearly explained to us about the difference between clinical study, clinical trial, definition of, say, investigational medicinal product, objectives of clinical trial, then current development in the area of this clinical trial, then cost involvement in the clinical trial, some historical aspect, then new development in clinical design, then career option in clinical trials. So in these 2 hours, you covered almost everything about clinical trial. You highlighted that clinical trials are conducted to collect data about both usual as well as unusual events associated with the use of investigational medicinal product. And this clinical trial needs to be well planned, well executed, and well reported, which can be possible through publication. So clinical research, utility, and here this clinical significance are 2 different things. So Dr. Van Dooren, we are fortunate to witness your session. Thank you very much.
Well, thank you very much, and thank you for this wonderful summary. I'm sorry it took me 1 hour and a half to explain everything, which you could do within 1 minute. So thank you.
Yeah.
So he's a best professor of pharmacology from our institutes.
Thank you, Satish sir.
Okay.
So once again, I thank you for your presence. I also thank Vice Chancellor, Pro Vice Chancellor of MIT-WPU School of Pharmacy, coordinator of this finishing school, and all student participants. Some students are there from Shivaji University, so I also would like to thank them for their active participation during question answer session and live feedback. I also thankful to coordinator for giving me this opportunity to host this session. I'm thankful to Mr. Pankaj Shirbate for providing technical assistance to this session. Now I request all the participants to switch on their video for group photograph.
That is needed for our records.
Yeah. Dr. Van Dooren, you please switch on your video for a minute.
Just have a beautiful picture for our records. Yes. I think Sir has not started his video.
Yes. Satish sir.
Yes, yeah. Sir has started. Yeah. I'll take snaps. It is recorded, not even. Yes, thank you, students.
Okay, so now you can switch off the video. I would like to request the coordinator of this finishing school, Dr. Satish Polshettiwar sir or Bhagwat Nagargoje sir, to announce tomorrow's session.
Yes, I'll announce. Yes, students, regarding tomorrow's session, as usual, it will be going to start at 10 a.m. with Saudamani ma'am. She is going to talk on employability, enhancement, skills and all. So you are supposed to join at sharp 10 o'clock morning tomorrow. Okay, so stay tuned with this finishing school. And thank you once again, Dr. Pawar sir, for a nice coordination and moderation of this session, as usual. Thank you, sir. Thank you.
Okay, so thank you all for attending this memorable session. I request technical team to conclude the session. Thank you very much, sir. Thank you, Dr. Dooren.
Thank you, Dr. Dooren, sir. Thank you very much.
Thank you, sir.
Namaste.
Yeah, namaste. Bye, sir.
Bye.